sort of strict about time. But every speaker is gonna have a slot of 15 minutes, ideally 10 for your presentations and 5 for questions. I'm not sure that the virtual attendees can hear that, but if you can ask the questions in the Q&A, that'll be great. For people here, you know, just stand here, raise your hand. Other than that, I think you're almost ready.
Check 1 2.
Thanks. Coming in.
Is it because we haven't started the show?
You can reply in the chat.
Okay.
Are we on? Are we on? Are we on? Are people online hearing?
Question and answer.
I can still hear the other thing, but that's one.
Yeah, it's low.
Okay, so just to recap, we're in the innovative imaging techniques for enhancing specimen-based discovery session. This is room C. And we're going to be starting our session with Jaimi Gray presenting Overcoming Large-Scale Challenges in Tomography, introducing resources from the Non-Clinical Tomography User Research Network, NoCTURN. Tilt this over the side so I understand behind this thing. Okay, so I'm here to talk to you about the Non-Clinical Tomography Users Research Network, also known as NoCTURN. It's not working.
Oh, because we stopped the. Okay.
Okay, now it's working. Okay, so NoCTURN is where the science of collecting and interpreting tomography data meets the nuts and bolts of good policies and best practices that ensure those data can be made widely available to a broad community. NoCTURN is led by principal investigators Jesse Mizell at the University of Texas, Austin, Morgan Chase at the American Museum of Natural History, Ed Stanley at the Florida Museum of Natural History, Brent Gillett at the University of Florida, and Paul Jidak at the University of Arizona. Also joined by our coordination and conferences chair, Leah Lynch, webmaster Dave Eady, and NoCTURN postdoc, and that would be me. So, but NoCTURN is so much more than just our core group. We have had over 200 people sign up for NoCTURN so far, and nocturnals, as we like to call ourselves, are spread across the entire US. We also have a few international members across the pond. So nocturnals are using data from many different kinds of CT scanners, including scanners from more than 10 different manufacturers, and we're scanning all kinds of different things. Most of these are biological objects, but we also scan geological, planetary sciences objects, industrial parts, batteries, fabrics, food, all kinds of things. And this data comes in more than 20 different file formats, and we're using more than 10 different software packages to process those files. So when you collect your data, and this goes for any kind of data, not just tomography data, you are the first curator of that data. And I want you to think for a second about what that means and whether it's enough to just throw the data up online and forget about it. So it's your responsibility to ensure that that data is fair and open, and that you provide enough accompanying information with that data to ensure that it's reusable. But we at NoCTURN know how difficult this is, maybe better than any. This is just an example of some of our collaborating labs that are part of the NoCTURN network. As you can see, there's a lot of different kinds of CT scanners, and trust me when I tell you, you'll have a very different user experience depending on which of these you operate. And this is just the hardware. This is not including the fact that you have to deal with the data, you have to deal with the metadata, you have to process the data, you have to store the data, you have to choose a repository. So when you begin or continue to deal with tomography data, you will begin to ask a lot of questions. And the questions on the screen are the kinds of questions that we at NoCTURN are trying to help you guys address by coming up with all kinds of different resources. And the way we do this is by having a bunch of different working groups. So what we have, we're made up of 3 different communities. This includes the education and outreach community, the basic and applied research community, and the industry and vendors community. Along with 4 working groups that represent the FAIR science principles: findability, accessibility, interoperability, and reuse. We also have an open science committee. We have online meetings regularly. Most of these working groups we meet once a month, and some of these working groups have even split off into smaller subcommittees that are working on particular tasks. We also have an in-person meeting every year. So last year we met at the University of Texas, Austin. This year we gathered at the University of Florida, and next year we'll be headed to the American Museum of Natural History in New York. Okay, but enough about us. I want to give you guys some information about the kinds of resources that we're generating. Starting with this, we just published our first of what will be several papers. So in this paper we illustrate how the standardization of data and metadata for reuse can foster interdisciplinary collaborations for large-scale data initiatives. So I encourage you to go and check it out. Now on to some of those questions that I was talking about. So let me walk you through a few scenarios here. So maybe you are just getting started with tomography data. Maybe you're confused about the terminology. Maybe you're not new to tomography data and you're still confused about the terminology and you want to brush up. So the accessibility and interoperability working groups can help you with this. We've been generating a resource which is basically a vocabulary resource. So on the NoCTURN website, you can find all of these different types of terminology associated with CT data that you might encounter, and how to make sense of those across different labs and across different scanners. Maybe you want to locate your closest CT scanning facility. Maybe you have some specimens that you want to CT scan and you want to locate the facility closest to them that you can send them to. Findability and accessibility can help you there. So in collaboration with our friends at MorphoSource, we have generated this world tomography map. So you can fly virtually around the world and you can find micro CT, veterinary, medical, or synchrotron machines that are all in the business of generating non-clinical tomography data. When you find a facility that you're interested in, you can select it and view the information for that facility and how to get in touch with them. Maybe you're trying to decide what scanner you should use for your particular project. Maybe you want to see the same object scan on different machines and see what the data set looks like. This is something the interoperability group has been working on. So at the University of Florida, we came up with this phantom or standard object. We made a whole bunch of them, and I have a few of them with me today if you'd like to take one home and throw it on your scanner. It is a bone produced by Lego, and we've drilled some holes in it. And inside of the bone, you have a silica nitride ball, an aluminium, a piece of aluminium, and a piece of titanium, and then an empty hole to represent it. So these are materials of different and, more importantly, known densities, so that we can put this object in different scanners and across different labs and see how the data sets compare. So this is what the setup looks like on our GE Phoenix system at the University of Florida, a scan that I did just last week. So I can record how I set up the scan, the different parameters that I chose, and then what the data set looks like. So on the left-hand side, you see the cross sections. On the right-hand side, you see the 3D rendering. And in the cross sections, you'll see those materials of different densities. And what I'd like to point out is that some of those high density materials produce artifacts. And we purposely did this because we want to see how the X-rays behave when they're exposed to high density materials across different tomography systems. And along with the data comes the metadata. It's really important to keep the metadata with your data. When you scan something, you make a lot of choices as a user, and we need to record all of these parameters so that we can replicate this across labs and systems. Okay, so maybe you're about to start collecting a whole bunch of tomography data. You want to come up with a plan to manage that data, and we have a couple of resources for that, produced by the Open Science Committee and the Basic and Applied Research Community. So the first is a data management and sharing plan. This contains some of the key points you'll need to consider when deciding how to curate and manage your data. This will soon be available on the NoCTURN website. The basic and applied research community has been working on a data decision tree. So when you acquire your data, that is just the first step. And then after that, you have to make a lot of decisions about the way that you handle the data, including how you process it, whether you are going to downsize or compress it, and how you do that, how you store data, and what repository to share it in. Speaking of repositories, maybe you have just collected a whole bunch of CT data. Maybe you have so much data that uploading the data sets one at a time just will not. So accessibility has been working on something for this, and in particular, Dr. Ed Stanley at the University of Florida, or the Florida Museum, has generated this code, which is available on GitHub, that will help you batch upload scans to the MorphoSource repository and save you tons of time with the data upload. Maybe you are publishing a paper that has tomography data, and you want to make sure that you correctly cite or acknowledge the lab where that data was produced. The basic and applied research community has been helping with this. So most of our associated facilities have come up with some kind of template that you can put in your acknowledgement sections or in your manuscripts that outline how you should cite that facility. And here's an example from our friends at UTCT. As you can see, it's just one sentence, and it really helps these facilities keep track of their data and the data's impact. Maybe you're about to publish your data and you want to make sure that it's reusable for the future. So findability and reuse have been working on resources for this. And what we've created is a document that outlines some of the information that you should include with your data set when you upload it that ensure other people can make the most use of it. Maybe you want to use tomography data in education outreach. The education outreach community has been really hard at work. So we've been gathering some educational data sets that we plan to turn into kits that we will distribute across schools across the US. And a few of those data sets include some plant data sets from our friends at the Danforth Institute, some mammal data sets from Leah Lynch at Winchester. Lost control. There we go. Some toy data sets, and then their accompanying real life counterparts. And then some food items, which include some coprolites with some remains of food items inside and some owl pellets. But the main kit that we're working on is based on some of my work. I produced these colored fallen models that represent homology across vertebrates, and we've been building an educational kit that goes along with these. The kit is still under construction, but the models themselves are available to view and download on my website. And lastly, maybe you, or someone that you know, much like me, loves tomography so much that you want to make yourself a career. And the Open Science Committee can help you with this. We've created a resource that outlines the different career pathways in tomography and the qualifications you'll need to go down those pathways. And I'll leave you with this. If you generate, use, or manage tomography data, you are welcome to join NoCTURN. You do not need to be experienced in tomography data to join. And in fact, we welcome people with all kinds of experience levels because that really helps us make these resources as accessible as possible. So you can join NoCTURN using the link on the screen, which I'll leave up there for now, so you can have it. And answer any questions. Thank you.
Thank you very much, Jaimi. Are there any questions?
Is there a plan for keeping all of these super helpful resources available after NoCTURN's NSF funding has ended?
Yes, there is, Julie. So when NoCTURN funding ends, so we 3 years of funding, NoCTURN is going to be folded into TSCA, which is the Tomography for Scientific Advancement group. So NoCTURN will continue to exist and it will be associated with the TSCA group. And so our website will stay active, you know, forever, hopefully. And we plan to make these resources available forever.
I just want to ask about the hardware. Is there a huge difference in terms of price of different scanners, and does that have a major impact on your decision
Yes, it does. So you've got quite a big difference in price. Some of the smaller like tabletop scanners are a lot cheaper than the big scanners that fill up an entire room. And then obviously you have things like synchrotrons, which are like billions of dollars. So we think that, well, we kind of hope that having the Phantom will help people make these kinds of decisions, because there is also, you know, as you go up in price, a difference in the quality. So there is a little bit of a difference between like a tabletop scanner versus, you know, a big one that fills up a whole room. And we just want to make sure people have all of the information to make those kinds of decisions. I think that answers your question.
Yes, thank you.
Thank you.
Wonderful talk. This is really a great resource to know about. I was wondering if there are any plans, even though you said that the NSF funding for it is going to be finished sooner rather than later, are there any plans to like host networking workshop kind of offerings, or even for, you know, new graduate students learning, trying to overcome the steep learning curve of what softwares to use, any workshops like
Yeah.
that in the future?
Yeah, so the facilities that are part of NoCTURN, individually, some of them do offer those kinds of workshops, and so we advertise those on the NoCTURN website. We have like a blog page with like news and stuff, so people can attend workshops. UTCT has a couple every year that people can go to, a lot of virtual workshops. I myself run a lot of working groups and things like that. So even, and then the other thing that we discussed in our last in-person meeting is that we're hoping, with our association with Tosca, maybe we can add an extra NoCTURN day onto the Tosca meetings where we can have some dedicated time for NoCTURN that sort of folds into that.
Okay, perfect.
Cool.
Thank you very much, Jaimi. We're gonna move along. So right now, we're gonna have Yann Pollet talking about Sphaeroptica, a tool for 3D taxonomy and geometric morphometrics in arthropods.
Yes. Wait, I need to. Share this.
Yann, can you share
Yeah.
your screen?
I'm right. Can you see it?
Yes, wait a second, our audio.
Is there a problem with my sound? Can you hear me?
It's on our end. Sorry about it. Give us a second.
Okay.
Where? Can you like go for it? I'll go find it. I can see and hear you virtually.
Yes.
Okay. Thank you. But we can have it. I think they're like out speakers. On Zoom. So it should be where the microphone button is. Where it says mute, there's a little arrow. So click that arrow and then it says select a speaker. So it might be a different one. Thank you.
That's real.
Cool.
What equipment if that makes it?
Okay, John, I think we're ready for you.
Yes, you can hear me?
Okay, hold on.
Hold on. Nope, not yet.
Okay, 2
Not
seconds.
yet.
Sorry?
Not quite yet. I'm not speaking.
I didn't understand.
John, can you try it?
Okay, yeah. Yes. Is it good? Is it working now?
Yes, yep.
Yep,
Perfect.
it's working.
Thank you very much. So hi everyone.
Put it on the
Um,
screen. Sorry, Yan, one more second.
Okay.
One more second. We're just technical difficulties here. Sorry, y'all.
It's just playing on the wrong screen.
It's because probably the PowerPoint is still. It's probably a PowerPoint.
Still sharing. No, we want to stop that from sharing.
It's because it's the PowerPoint that's probably running on the background.
Oh, here we go.
No, it's
Fine.
a closed PowerPoint. So it's like you have to minimize Zoom.
Okay, so yeah, close the PowerPoint. I think is what she's doing. Want to close it or just figure out the.
Close them and turn them. Yeah, I don't know why it's sharing. Maybe that was the same. Okay.
That one dominating our screen or something. Is that him?
It was not showing.
Can we go full screen with this?
We need to get like his onto that author.
So what's up, everyone? Sorry about a couple of speakers dropped out of session, so we'll probably be ending on time for you to get a call.
That swap video with zero screen that didn't work. Is this his PowerPoint? Is that his power?
Yeah, but he's sharing his screen on Zoom.
Right, so, but is this his
Yes,
PowerPoint?
yeah.
That's him.
Yeah.
Yes,
Okay, so that, oh, but we
but
can,
we can see him. Yeah. People online are seeing him, but we're not seeing him
Right.
together. We can only see the PowerPoint now that he is sharing his screen. The speaker video is on the podium.
yeah, I'm not talking, so maybe that's why
you're gonna put it right now.
you have the sound that's showing the podium.
Mm.
What's a quick solution?
Okay, they said that when we start talking on the mic, it changed to the podium view.
Yeah, I guess it's normal. I guess it's normal.
We cannot, oh, there it is.
Is there now?
You can just exit out of the chat for the duration of this talk. There we go.
Okay. Just click OK for that.
Yeah, just click OK. I can hear you.
Yes.
Okay.
How can we like?
Get rid of that.
Yeah.
So will you
Mute it. Oh, wait, wait.
Yeah,
So
This is on the one that's going out, so that's just yours.
I would just keep it for now. Just keep it simple. Is what I would say.
and I'm gonna mute us.
Yeah.
Can I start?
Ask him to talk.
Hey, can
Yeah.
you talk?
Yes.
Yann?
Yes. Do you hear me correctly?
So go to your microphone, hit
Yes.
the up and see where the output. So it's going out to headphones right now. So you need it to go out to the speakers.
That's what it's going out to the speaker through
Oh,
through the headphone.
Okay. Sorry.
I don't know why he stopped. We had him.
Yeah, we have.
Oh, you
Uh,
did have
had
it.
him.
Can you hear me?
Uh,
Can you hear me
oh.
correctly?
Here
Yes,
now. Thank you
yes.
so much.
Yes,
Okay. Is it working on in the room as well?
Yes, it is working in the room. You are loud. and
Okay.
clear.
Perfect. Okay. Perfect. So I can start. Um.
Okay.
So hi everyone. Digitization of natural science collection has, for the past decades, been an important challenge for scientific institutes. And in this regard, we developed Sphaeroptica, a new 3D digitization tool specifically useful for small specimens with complex structures, as arthropods. It allows taxonomy studies and morphometric measurements on virtual specimens without the need of expensive equipment. Digitization is important in many ways. To list a few of them, it allows some studies that were impossible before, and it allows an easy access of collection to scientifics around the world. For the latter point, the EU has funded different projects, like the Synthesis series, to simplify the access of scientific collections between institutes inside the Union. Under this project, the Royal Belgian Institute of Natural Science tested a lot of digitization techniques to find the adequate ones, depending on the type of specimen and the type of digitization needed. We have published a handbook of best practice for Synthesis 3, and we have created the application DigitKey, where multiple European institutes present the setups they have. If we focus on small specimens, we can see that they usually have complex and small structures, such as setae and antennas for arthropods. And capturing all that information will be important for further studies. Therefore, having a setup that gets high-resolution images through focus stacking around the specimen is required for its 3D digitization. For a complete comparison, we decided to test two alternative setups at the same time, MicroCT and Structure Light. One of the main setups for photogrammetry scanning of pinned specimens is a D3D scanner created by the Digital Archive Natural History of Darmstadt, as it is also used by many institutes in Europe. It acquires automatically focus-stacked images on many views around it. Then, this can be put in a photogrammetry software to create a 3D model. Before explaining how Sphaeroptica works, let's see what setup we used. We chose to build a scan device for the image acquisition. It is an open hardware alternative to this 3D. We chose this solution because affordability was a crucial aspect to us. Scanned also has a focus stacking process that we have used for the tests. However, with further tests, using other softwares can lead to better results. And lastly, for the calibration, we decided to go with the commercial photogrammetry software Agisoft Metashape because we were already familiar to it. Note that open source alternatives as CalMap will work as well. Sphaeroptica is based on one specific motto: less is more. So after getting high resolution images and calibrating them, instead of asking the computer to make more computation to create a 3D model that can be suboptimal, Sphaeroptica use that data directly for their built-in 3D viewer and to compute 3D landmarks for measurements. We have a quick demo of the application. So the first functionality is the 3D viewer that displays 2D images in order to see them in 3D. On the top right, you can see the shortcuts that get designated views. Just under, there's a list of landmarks. It's customizable. You can change the color, change the label, change the position, delete them, and also reset them if a position is incorrect. And to place a landmark in the 3D world, you need to place on a picture on at least 2 times. This will trigger an algorithm called triangulation, a basic principles of photogrammetry. The blue circle you can see on the right is a projection of the blue landmark wing L1 on this image. And then the last functionality is that we have 3D compute. We have a quick distance calculator, sorry, that computes the distance of 2 3D landmarks, and we can scale the entire model with it. Lastly, it is possible to export all the points in a CSV format file in case you need order computation that Sphaeroptica doesn't do. Now let's compare Sphaeroptica to the other digitization techniques, microCT, structural light, and structure for motion. We use 4 different case studies for it. The Euphorus, that is quite simple at spruce. The Ovalicia, that's bright. The Lycus, that's a flat specimen, and the Phylodicus, a hairy specimen with transparent wings. And the first comparison is visual, to see which models are good enough for taxonomy studies. Here, we can see that the microCT model is much more detailed than the other ones. Unfortunately, we don't have the texture, and the metallic pig creates some artifacts on their model. On the other side, the structural light and structure for motion models are too smooth to be used. A lot of important info for taxonomy studies, especially the CTs, are missing. And finally, we have Sphaeroptica that has the highest resolution possible, but doesn't have a 3D model. Will it be a problem when it comes to measurements? To answer that, let's compare the accuracy of Sphaeroptica to the other solution. And because microCT has shown to be extremely precise, we will consider their measurements as true. The results show that Sphaeroptica usually gives measurements with less than 1% of error. The only exception here is for the Euphorus, a bigger arthropod that needed more images for the focus type. And this added a higher distortion to the picture, thus a higher risk of error due to noise. Lastly, let's compare the time that takes each workflow to see their variability in a mass scale digitization program. Sphaeroptica and structure for motion workflows may take the longest time at around 2 hours and 30 minutes, even after optimizing the focus tracking process by a factor of 12. But this is a time where the scientist isn't bound to the digitization and can work on something else. When we compare that to microCT, where the process may take only 30 minutes, but someone has to work on it during half that time. On this particular point, the structural light workflow is the best workflow, as it is faster than photogrammetry and doesn't require as much human input as microCT. Unfortunately, as said before, the resulting model isn't optimal for scientific studies. Now, let's summarize what we've learned. Sphaeroptica has shown to be precise enough for geometric studies and has also shown to be the best solution for taxonomy studies. However, it is dependent on 2 things, the quality of the images and the quality of their calibration. Therefore, optimizing these will be crucial for the solution. At this time, we are working on a second version of the app, using web technologies as front-end, meaning that we can deploy Sphaeroptica in many more ways, web app or desktop app, for example, than we can do right now with Qt, where it's only a desktop app. And the main reason of this change is the fact that we are trying to place Sphaeroptica as a plugin of Orthonc. Orthonc is originally an open source server specifically made for medical imaging. And as of right now, our institute is working to implement it to store their digitization data. The server is open source, scalable, and allows developers to create new plugins for their needs, which we are trying to do with Sphaeroptica. We are at the end of this presentation. The code of Sphaeroptica is completely open source, and you can see it on GitHub under this link. And if you have any questions, I'm here to hear them.
Thank you very much, Yann. We have time for questions if people have any. Nope. Okay. Yann, thank you very much for your talk.
Thank you very much.
Okay, we're gonna move on with Fiorella Esquivel talking about entomology meets engineering, deciphering the workings of the bee stinger and beyond.
Hi. Let's see if I can get this working. Oh. How's that looking? Can you hear me?
Looks good on our end. Yep.
Yep. Okay, great. Awesome. All right, I guess I'll get started then. So, hi everyone, my name is Fiorella. It's 2 a.m. in Australia, so I'll try to be coherent, but no promises. So today I wanted to speak a little bit about a workflow that we've come up with for working with microcity data and doing sort of biomechanics assessments of insect systems. So the system we're working with is obviously the bee stinger, but it can be applicable to other systems as well. So our case study is a bee stinger, so let's learn a little bit about that. So many of you have probably been stung at some point or another by a bee, and if you have, my condolences. It's pretty unpleasant, but you may have noticed that bees have the ability to do this pretty nifty thing. They can leave their stinger behind to continue to sting you once they've left. And we call this autotomy, and it's a bit of a unique adaptation. So this is a sequence of images from an older publication showing the stinger being left behind. And this is done due to some rearward-facing barbs, which make it difficult to remove from the vertebrate skin. They look a little bit like harpoon barbs. Oh, it looks like it's another timer. That's delightful. Anyway, so the barbs make it difficult to remove from elastic vertebrate skin, and the other nifty thing that they do is that muscular contractions continue after autotomy, and this helps the stinger propagate into tissue and pump the venom. So the other thing that comes along with the stinger is the glands, the pheromone glands continue to secrete pheromones, and they recruit further bees to the site and get other bees to sting you as well. So that's pretty fun. So when it comes to autotomy, there are some very clear costs to this. This can damage the lower digestive tract of the bee and lead to worker death. But the benefit is that the stinger continues to sting regardless of whether the bee is brushed away or killed or removed. And this is pretty important for the bee, as its reproductive success is tied to the reproductive success of the hive. One of the challenging things about this adaptation is that the stinger needs to pack everything that's needed for stinging into a very small package. And to give you an idea of the size we're talking about, this is a stinger on my fingertip. So it's pretty small. So we wanted to understand how it works a bit better. So we needed to understand a little bit about the anatomy. So there were some pretty thorough descriptions in the past, but they look a little bit like this. It's a bit hard to make heads or tails of what's happening in 3 dimensions. So we decided to do some micro-CT scanning to understand the 3D morphology. And we also wanted to know about the kinematics. So we did some high-speed filming, and I'll get into that in a minute. So very quickly, I worked on dissected stingers, and I dyed them with leukothioidin, so nothing too fancy, and we did the scans at the Australian National University. So After that, we got something that looks a bit like this. It's a fairly complex organ, so I'll just walk you through it slowly. So we've got several components, including glandular materials, as I alluded to earlier. So that includes the venom sac, which stores the venom. There's also neural components. There's a neural ganglion that coordinates, oh my goodness, that coordinates sensory information and muscle contractions. And there's also obviously the muscles that pump the venom, and the cuticular components, which is a main area of interest today. So that's what pierces into the tissue, pumps the venom, and supports the musculature. So focusing on the cuticular components, if we have the stinger and we remove this extraneous materials, we get this venom sac, which empties venom into this bulb structure, which is the larger structure below. If we remove these two things, we get the lancet. So these are two mirror image components, and this is where all the action is at. So on one end of the lancet, we have the piercing part, which pierces into the tissue. Then we have the valves, which pump the venom. And on the other end, we have articulation with the plates and the muscles that power the whole system. So piercing and pumping have to happen concurrently because everything's located in this one piece of cuticle. Another way to look at this is we're just looking at a side angle. This is top down. So we have the stylet, which widens into the bulb to receive the venom, and the lancets are connected to this via a series of rails, which we call the oscillator. So the lancets slide backwards and forwards along these rails relative to the stylet. The three pieces, the stylet and the two lancets, come together to form a venom canal. So it's a composite structure. But that doesn't answer how the stinger pumps the fluid. So if we took an optical section through here, remove the back, we get something that looks like this. So we're turning it towards the screen. So we have these two valves that we have mentioned before, pump the venom. So diagrammatically, we have an inlet at the top, outlet at the bottom, and these two valves. So this is a bit of a weird way to make a pump, but I'll get into it a little bit more. So to find out how this works, we did some high-speed videography, and we get something that looks like this. So this is the bee stinger going into some silicon substrate and pumping venom. So as it's working its way further in, you can see this little venom droplet accumulating on the surface, and it's pumping the venom with these two moving structures in the middle, which are the valves we looked at before. So from this, we can see that these double valves are pumping in antiphase, and because the pumping and the piercing are coupled, if we digitize one, we know the kinematics for both. So we can get some nifty graphs like this, and you can see both valves moving in opposite directions. And if you squint a bit hard at this video, you can also see that the valves are changing shape as they're pumping. So they're collapsing on the upstroke and opening up on the downstroke, but there are some limitations to our resolution here. So we decided to move to an artificial system to study this a bit more closely. This video that I'm showing here is filming through the stinger. So I'm shining a lot of light through it and filming at 100 frames per second. So it's a little bit grainy, you can't fully see what's happening. So to study the pattern of fluid flow and the deformation, we moved to 3D printing. So we took our 3D reconstructions and designed an artificial pumping system that mimics the shapes that we see in the stinger. So it's a little bit simplified for manufacturing, but it conserves the inlet and the outlet and the basic outlet, layout of things. We did have to go through quite a lot of iterations to get to this. So we needed something that could handle air and fluid leaks, could house actuation motors and Reynolds scaling. So the whole experiment is done using glycerin to replicate the Reynolds scaling. Went through a lot of different designs. We also simplified the valves to make them something we could manufacture using flexible materials with 3D printing, and tested a lot of different plastics to make sure we could find something that could do what we wanted. But we were successful, and we got something that looks a little bit like this. So this is an artificial rig pumping. So it's filmed at 1,000 frames per second, so it looks a little bit odd and funny, but this is very exciting. We finally got something that's working, and so the next steps are to try to visualize the flow a little bit better and see how the venom is traveling through the bulb and like what vortices and interesting things are happening in there. But we're getting the valves to open and close correctly, so very exciting stuff. So stay tuned for further developments in this area, but very, very happy with this. We are also working on an artificial piercing system that mimics the reciprocation of the piercing component. So we have a student working on this, just mentioning briefly, but for today's purposes, won't go into too much detail, but again, using 3D printing based on the CT scans to replicate the piercing structures. Now, the exciting part is now we're looking at trying to use the same pipeline to study a range of different systems. So the Hymenoptera, so bees, ants, wasps have really quite diverse adaptations in how they use their ovipositors and stingers. They can use them for predation or carrying prey or like wood, boring into wood, doing very delicate sort of surgical precision, like stinging into brains and stuff like that. So we're interested in looking at a range of different species. So we're collaborating with the National Insect Collection here to sample throughout the Hymenopteran phylogeny and try to identify some morphological adaptations for different ecological niches that we can look at using the same kind of pipeline to study biomechanics. Yeah, so stay tuned in the future for more stingers and ovipositors. And with that, I'd like to thank all of these people, and especially Nicole Fisher for encouraging me to come and present at this forum. Yeah, thank you very much.
Thank you very much, Fiorella. Do we have any questions? We do have questions.
Hi, nice talk, and props to you for being up at 2 a.m. I know the time difference is really hard. So with your 3D printing, you mentioned you tested a lot of different materials. It looks like there was a little bit of flexibility in the one you ended up with. I was just wondering what you ended up using.
Yeah, so we tested a whole bunch of different TPEs and TPUs. So they're like thermoplastic urethanes, I think. So they were all flexible materials, and that's why we needed to do so much testing with them, because they all have different elasticities, and there are different factors in how they behave. So we wanted something that had a little bit of spring, so that we have lots of plastics that could deform, but they wouldn't necessarily go back to the original shape in the way that we wanted them to. So that was a bit challenging, yeah.
Have another question.
Answer the question?
Hey, do you foresee any biomimicry applications for these findings?
Yeah, that is actually the intention. So one of the motivations is to try to see if we can use learnings from this pump design to design like medical devices or other types of micro pumps. It is a really unique way to pump fluid, but it is also fairly heavily reliant on the viscosity of the fluid, because it is such a small little pump. Viscous effects dominate, and so it would be best suited for something either very small or some very viscous.
Any other questions? I think that's all that we got for questions for you, Fiorella. Thank you very much.
No worries. Thank you.
Okay, next speaker is Julie Winchester talking about MorphoSource 3D data repository, new features and capabilities empowering media contributors and consumers.
Right, for advancing slides.
I'm trying. Nope.
Yeah. Hey, everybody. So I am Julie Winchester. I am while they figure things out, I'm the technical director for, the MorphoSource 3D data repository. We good?
Yeah.
Awesome. Thank you so much.
Point it towards here.
Oh, yeah. Got it. Cool. And Yeah, so, and I will also say, just coming in the room is actually my colleague and collaborator, Doug Boyer, the founder and director of MorphoSource, and I will make a plug. He's gonna give a talk tomorrow morning about MorphoSource's sustainability model. It'll be a very cool talk. Recommend to go see it. All right, so what am I gonna talk about though? I am going to briefly introduce what is MorphoSource, for people that may not know about it, and then after that, I'm going to do like a real quick whirlwind for people who have used the site, just to talk about some new features that we've added in the last year. But I'll keep it high level, I'll keep it brisk. And then at the end, I'm going to talk about a feature that we are currently working on, which we're really excited about, has real potentials to really like level up how we actually store data behind the scenes. So, but what is MorphoSource? I kind of like this elevator description that MorphoSource is a digital repository service where researchers, museum curators, and the general public can find, download, and upload 3D, 2D, and audiovisual media representing physical objects of academic or scholarly interest. Some more digital creations, and a lot of those are biological specimens. They are natural history specimens from biodiversity collections. So that's the one sentence, what is MorphoSource? To give you more detail, yeah, so it's a free digital repository. The metadata and the features are 3D specialized, but we do support 2D images, video, that kind of stuff. Yeah, data mostly represent physical objects of scholarly interest, which is a long-winded thing to say, mainly natural history specimens. And the data are not contributed by us. They are contributed and managed by a community of users. We don't, own any of the data in the system. It's a system for helping contributors share their data with other researchers, with the public, with the world. Point it that way, keep getting.
Sorry, Julie.
No worries.
People are not seeing the PowerPoint online.
Okay. Yeah, I can. Yeah, so different things that you can do with the site. For most users, you may interact with MorphoSource as a data discovery platform. There are various browse and search tools that you can use to find data contributed by other users, and then you can download those data, which some of the data are downloaded openly. You just tell a little bit about what your usage information is, and then some of them are what we call restricted download, where you actually have to fill in a little request. You're like, I'm going to use these for my PhD dissertation. And then the contributor logs into MorphoSource and actually sees that they have a request in their system, and they say, oh, that's great. As a contributor, you can use the site to upload data, but you can also use it to manage and track data. So I can upload my data, I can see how many times it's been downloaded. As I said, we collect usage information, so I can see that 80% of my usage has been for research, but maybe 15% has been for art. And maybe I'm a different person, and 30% of my data 12 education. So that's the level that we can parse those things at. One of our user communities also are object collections and imaging facilities, and they can use the site to track the usage of their things and to manage media. So oh, there we go. Awesome. Yeah. Cool. So basic facts, figures about the history. We have actually celebrated our 10-year anniversary. It was founded in 2013, and organizationally it's part of Duke University, but it is funded by NSF grants, Duke commitments, and by some data contributors who have large data sets, who have grant funding for data storage. We are able to have those users support the site. We basically rebuilt the whole site in 2021, and since then I've got a little count on the right side. You can see we have almost 20,000 3D media representing almost 70,000 physical objects. And we collaborate with a lot of different museums, institutions, standards groups, working groups to sort of follow and develop data standards. Like we're working very heavily with NoCTURN that Jaimi talked about earlier to try and push data standards further. So the real nitty gritty, like here's the media types that we support: 3D meshes, 2D images, video, but also image stacks and collections. So that might be an image stack for a CT scan, image stacks for photogrammetry, or histological slide sections. We support all of those. And we support a very long list of imaging modalities. You'll see that some of these are underlined. Those modalities, we have modality-specific metadata. Probably most of that metadata is going to be for the CT, but yeah, but there are others. And things that we really put a lot of time working on in the repository is, yeah, Jaimi mentioned these FAIR principles: findable, accessible, interoperable, and reusable. And the site is really built to uphold those. We have some best practice guidelines for formatting and file type. We generally only accept open file formats, things that we think are going to have long-term file preservation potential. And we also publish our data model and our terms vocabulary. We actually make reference to community standards like Darwin Core or for CT DICOM whenever possible, but we do publish those. They're out there. You can sort of look at the data model. And this is that data model. It's really, but the whole point is that we have a strong focus on letting users document imaging and processing workflows, which, for 3D, if you work in 3D, can be super complicated. It can involve 10 different applications and 20 different steps. And a lot of the emphasis in MorphoSource's metadata is in letting you document those sometimes complex workflows through your imaging events, through your processing events that have metadata at each of those steps. That's a big thing we focus on. And it is an actively used site. So you can see here, media downloads on the left since 2016 and media views on the right since 2021. So the time horizon is different for these because in 2021, we changed how we measure views. Some interesting things that you can see here, an obvious pandemic bump as everyone was trapped in their home and everyone had to, it really showed the potential and the power of digital data for allowing people to work wherever they are. And of course, also notice there's a bit of a bump in this last quarter, and that is in part thanks to OVERT. So OVERT came out, made over 20,000 scans of vertebrate specimens, and they've had their big paper, and it's out there. Though I will say it's the media, the OVERT media is important, but it's less than maybe 15% of the stuff on MorphoSource. So there's a lot more stuff than just what OVERT scanned. Cool. All right, so that's the what is MorphoSource talk. I'm just going to go through real quick some features we've added last year. All right, so let's say I've got a private media record. I'm waiting to publish it. I'm not ready to let someone access it, but I want to send it to a journal. I have an article in submission, and a reviewer wants to see it. One of the things we added are anonymous access links, so I can take my media, I can generate an anonymous link there, which for anyone who's really eagle-eyed, I've already deleted that link, so you can't use it to get to this media. But and then the journal reviewer can then use that link to access the media without needing an account on the site. They can see the private media, and you can actually do that for an entire project. So if you have multiple media, a reviewer can take a look at them through that. We've added a new way of grouping media, a media list. It's like a playlist. It's like Spotify or a YouTube playlist for media. Should get a minute or two longer.
I gave you a minute extra.
Yeah, so basically anybody can create this. You can add any published media on the site. This is one that I threw together a few weeks ago, just of collecting some various platypus scans. You can actually see one of Jaimi's colored homology forelimbs if you want to see a cool platypus forelimb. Data vocabulary, I mentioned this, but it is out there. It is human and machine readable. It's in OWL format. The API, we've done a lot of cool stuff. If you're like a technical person, we've done a lot of expansions to the API. You can actually now download media entirely through the API. Some of this was motivated. Murat Maga's group and collaborators have actually created a Python package for interacting with the API. So check that out. And remote media. So sometimes we have institutions that want to have media in a remote server. And so they're like, well, I don't want to, I don't need this stored on MorphoSource, but I'd like to have the MorphoSource discovery tools. We can now actually support media in a remote server with a metadata-only record in MorphoSource. If the server is set up right, users can even download that media where we stream it from the remote server. And unfortunately, I don't think I have enough time to show you our viewer improvements. But we're gonna make some really cool viewer improvements soon. We're gonna add orthographic cameras, lighting controls, all kinds of cool nifty stuff. Yeah, collections facilities, mainly just because I know I'm running out of time here. So we have this concept of organization, which is just any autonomous bureaucratic unit. This is what we use. Sometimes entire museums manage their stuff together. Sometimes it's on a level of collection. So whenever I say organization, it's an imaging facility, it's a lab, it's a collection, it's a museum. And collections and facilities can use MorphoSource. Staff members can directly use MorphoSource to manage and track their stuff in MorphoSource, including the ability to view all the stuff either that was imaged at your facility or that represents one of your specimens. So for collections, yeah, collections can also provide recommended ownership settings. They can serve as long-term managers of published media. If you are already doing this, you may have received an email for us. We're simplifying it significantly. We're getting rid of teams, but if you're not familiar with it, don't worry about it because they're going away. And facilities, we just added a whole bunch of new features to enable facility members to track media that were imaged at their facility and to edit and create device records. Okay, I'm gonna go real quick. Yeah, so storage is expensive, right? Everybody knows storage is expensive. What do we do? What do we do to fix that? So right now, all of our data is in hot storage. It lives in file system storage, like this one on the left. It's in directories. But there are other storage models. There's object storage, which is sort of like you have a digital bucket. You put stuff in the bucket. It has an ID that you associate with it. And then you don't really care where the files get stored, so it can be a lot cheaper. But there's also cold storage and cloud suppliers. Right now, all of our data is on-prem at Duke, which has advantages, but maybe we want to use cloud as well. Thankfully, Duke OIT was recently funded by NSF to build a big fancy storage cluster that basically blows open the doors and lets us store data across all of these. We can move stuff file to object storage. We can move stuff hot to cold. So what are we doing in the next few years? First, we are moving all of our stuff to object storage, which instantly makes all of our storage cheaper. Then we are implementing the ability to move data between hot and cold storage tiers. But the really fancy thing is that we are then adding an all-seeing algorithmic eye to manage these things. Basically, we've come up with a survivorship model that allows us to put probabilities on how long it'll be until a data set is downloaded. So if we can say I have a 99% probability that this media will not be downloaded in 2 years, that's something we can move to cold storage. And then we're going to have a system where users will be able to retrieve the data from cold storage with maybe a bit of a wait. You know, like your data media will be available within 24 hours. And altogether, we think that this is going to make for a significant savings on data storage costs for the long term. That's what I've got. Hopefully we have time for questions. Thank you.
We do have time for questions.
Thanks, that was really fascinating. I came in after the beginning of your talk, so I don't know if you talked about this or not, but I'm curious about the way that you guys may be preparing your data to make it accessible for people using computer vision and trying to make the imagery available for, like, you know, AI ML scientists and things like that.
Yeah, no, it's a good question. So we have not directly to date worked with AI computer vision stuff, but I think we have a lot of stuff that sort of sets the landscape for it. For instance, every media that comes into the system, we run a whole barrage of automated file processing so that we're able to pull out, say, machine readable metadata for the file. So it's not just what the user inputs. We're able to take out a lot of that, but also we actually have a pretty complicated system of creating low-res preview assets for things, which can sometimes take, I mean, we'll take a 20 gigabyte CT scan and we'll shrink it down into like a 30 megabyte sort of web preview of the image stack. And then we make that preview accessible through standards, except like standards pathways, like all of our preview assets are available through IIIF manifest, which is for anyone, if people aren't aware, it's like used very commonly for libraries and museums as a way to interchange image information. So a lot of this stuff is, and again, we have the API and everything, so a lot of it's like well set up for that. And the ability to have a preview tells you that the data is solid, that if a computer can read it, it can work with it. Does that get a way to answer your question?
I think we're out of time for questions, but feel free to talk to any of the panelists after their talk.
Thanks, everybody.
Okay, next up, we have William Weaver with the talk Exploring the AI Kaleidoscope with VoucherVision, a spectrum of techniques for specimen label transcription.
Okay. How did you get it to work before? No. Share screen. So close, we're so close.
Thank you for everyone's patience.
Really appreciate your heroic effort. Screen share. Oh, wait. Okay, it's not screen this. Yeah, good.
Right. No. Wait, is it no, not yet. So sorry.
That's not it?
How do I share this?
It is shared.
Well, but it was the presentation mode. Oh my God.
We have one more presenter after it. It's virtual, so we're gonna get it down just at the end. It's
Okay.
not the screen too,
Why not?
because she specifically said to do the.
Oh, why is it open 3 times?
You know, that's it.
Because they're seeing
We're
the
seeing the speaker view, you're right. So the other. Yeah. So you
Can we make it not speaker view?
like screen switching. Okay, so let's try the other. Sorry. So try. Doesn't. I'm just going to share it. So let's try starting screen sharing. Presentation. What do you see? A single. That's.
I'm so sorry.
It's not. It's not. I'm not sure. I'm afraid I'm gonna see that. Screen. Yes, yeah, yeah. Yeah, okay.
You're good. Is it working now?
Yes. Great. Okay. Thanks, Chanel. Good morning, everyone. My name is Will Weaver, and I'm from the University of Michigan. Today I'll be talking about some of the projects that we are working on to hopefully use AI, whatever that means, to increase the pace and efficiency of museum label transcription. So this is the basic overview for today's talk. We'll be discussing a few of the tools we're making, including briefly touching on Leaf Machine 2, VoucherVision. I'll discuss our efforts to make custom curatorial large language models. And then we'll wrap up by, again, shortly talking about quality control for label transcription. So if you are unfamiliar with herbarium specimens, I just wanted to give a little bit of background of what they look like. So much like libraries, we have stacks that are filled with preserved plant vouchers. They're typically organized by taxonomy, and an average voucher might look something like the middle image. There will be some kind of plant material that has been pressed and mounted along with archival information.
Will, can you talk closer to the mic?
Sure. Yes, along with some kind of archival information. And herbarium specimens are widely used in biological research. They are useful for their historical record. We can obtain genetic information from them, and they can inform changes in biodiversity, such as the spread of invasive species, and they can help us inform conservation status. So a lot of my research has focused on the evolution of leaf shape in flowering plants. So we've extensively used herbarium specimen images to extract quantitative trait data, such as leaf outlines, leaf area, perimeter, things like that. And to do that, we've been developing a tool called Leaf Machine 2, which leverages an array of machine learning algorithms to extract quantitative trait information from digitized herbarium specimens. And while this isn't the focus of today's talk, I just wanted to say that since publication, we've added quite a few updates and trained all of our algorithms on about 5 times as much training data as was in our publication. And we've been adding little tools to help take advantage of all of the metadata that Leaf Machine 2 produces. So one example is the ability to censor archival components in an image, which is useful for image classification tasks, for example. So if you want to learn more about Leaf Machine 2, you can go to leafmachine.org and read all about it. So the ability to conduct this type of research is the direct result of decades of extensive effort in collections to digitize everything. And this digitization process has many expensive steps. The specimen has to be mounted to an archival sheet, has to be accessioned, has to be imaged, labels have to be transcribed, and the specimen must be curated and its placement periodically updated when there is a taxonomic revision. Of all of these steps, one of the most costly and one that contributes heavily to our digitization bottleneck is the label transcription itself. So for the images to be fully databased, someone has to go in and manually transcribe all of the text that's in those labels. And that's the portion of, that's what we'll be focusing on for the rest of today's talk. So before we discuss transcription, it's also helpful to take a look at what kind of information we have to transcribe. So on the left, we have a collection of images. The middle an example of a typical specimen information, like the collection stamp or outdated determination. So looking at a few more label plots, we can also see that there's a huge diversity of quality and readability depending on the age of the specimen. Modern labels are usually typed out, while other specimens that are older are usually handwritten, often in cursive, and they're difficult to read. They're also cluttered. If it's 200 years old, there's a good chance that the taxonomy has been revised a handful of times along the way. So for current manual transcription process, a curator can probably manage between 15 and 20 specimens per hour if everything is going smoothly. And the most time-consuming process is when a curator has to go to an external data source to validate some kind of data component. So that usually means going to Google and searching for something. With older labels, you can imagine that if something is 100 years old, it could be non-trivial to figure out where a geography from 100 years ago translates into a modern geography. And to put all of this into context, only about 48 million specimens have been digitized globally to date, and that's out of a pool of probably at least 330 million specimens. So if we do some napkin math, it's easy to assume that we're decades from completing this digitization task. So any sort of tool that can help us increase the pace and the accuracy and the speed and the efficiency of this process is invaluable. And critically, all of these records need to be findable and accessible in some sort of database. Right now, anything that hasn't been digitized is basically unknown to the world. So it would be great if we could get all of those added to make them searchable. So that neatly brings us to a project that we've been working on called VoucherVision, which is designed to be one of those tools. The goal of VoucherVision is to use optical character recognition and large language models to extract and then parse the text into an appropriate field, so a JSON dictionary, and then eventually into just a basic spreadsheet. So as an overview for VoucherVision, we begin with the full specimen image, and from there we use Leaf Machine 2 to extract just the label information, and we send those label collage images to the OCR engine. And then we can begin to assemble a prompt that we will submit to the large language model. So if you've used ChatGPT, it's basically the same thing. We're just doing it through an API, and we're dynamically assembling those things. So the instructions basically contain guidance for the large language model, including specific formatting instructions. And these instructions are based on the same guidelines that we give to our curators when we're manually transcribing the images. After that, we just show the large language model the unformatted OCR text. And then we showed an example of what we expect it to give us. So we basically give it an empty JSON dictionary with all of the fields that we want it to transcribe. And then finally, more granular rules for each of those. That prompt is submitted to whatever large language model you want to use. So that could be OpenAI's GPT-4o, Google's Gemini, or any of the other open source large language models. That will return a JSON candidate. Then we have some other validation steps to make sure that our JSON dictionary is correctly formatted and that has everything that we need it to have. If it fails, we just send it right back into the large language model with a different instruction to correct that mistake. From there, we have a JSON dictionary, which is basically just a row in a spreadsheet, and that gets, you can send that into whatever database you'd like. So that describes one of at least 4 implementations that we are currently have implemented in VoucherVision. So what I described can be referred to as prompt engineering, where the prompt includes all of the necessary instructions and context for the large language model to accomplish a task. And for VoucherVision, we've been testing many different ways of doing this, such as really short, simple prompts with single or multiple instances of OCR, and then all the way up to very detailed thousands of words long prompts that all end up giving you something in return. So the goal is to figure out what's the best method of accomplishing all of this. And a benefit to prompt engineering is that it's relatively straightforward. It's pretty easy to see what's going on, and it's easy to see what sort of changes are affected. So a second approach is to fine-tune your own large language models. So that involves creating a training data set and then teaching an open source large language model to do one really specific task. The benefit of that here is that instead of a 2,000 word long prompt, all you do is show it the OCR text, and it only returns the JSON object. So this is much more efficient. It's really customizable. So this is also a really good option. A third approach, which is really cool, is using AI agents. So in this case, we have a supervisor large language model that sends tasks to smaller worker large language models. It receives information back, and then it self-corrects. So this is really cool. You can even watch it fixing mistakes. And the benefit is you get really high quality transcriptions as a result, and very few mistakes. And then finally, we can do all of this in a single step. I don't know if anyone's played around with it, but the newest OpenAI models are pretty insane, and they're really good at reading handwriting. So one thing you can do is just do all of what I described in a single step. You give it a really long prompt, you show it the picture, and you get back the JSON object. So I just have some examples of how it's pretty impressive. I instructed it to put any long form text into the occurrence record, and it basically did it perfectly for this example. And it basically got this one completely correct also. So each of these is beneficial for different collections purposes. There's a lot of different pros and cons. There are locally run open source models, which are basically free if you already have the computational power to run them. There's also paid solutions. The paid solutions are generally going to be more advanced, but they're also much more expensive. But there are some cheaper options too. So for example, the single step version using GPC 4o is extremely expensive. It's about $20 per 1,000 specimens. If you were to use that for the AI agents approach, it would be probably 5 times that. So there are very expensive ways to do this, but with VoucherVision, we're also working to make sure that we have completely local solutions that are much more in line with the budgets that all of us have. So right now in VoucherVision, we have these 4 supported workflows in mind. We could envision using a local-only prompt engineering approach for general use cases. You could run local-only fine-tuned models to do some other really specific tasks. We imagine using the single-step approach for really complicated handwritten labels. So anytime you have really old labels, for example, you could just push those into a different workflow. And then the prompt engineering with APIs, some of those we can engineer to run extremely fast. So we can call the API in parallel. So you can use VoucherVision to process something like 1,500 images per minute, which is pretty insane. And at the University of Michigan, we're planning to use that to create skeleton records for a huge pool of images that are just sitting there and make those at least discovered. All right, so I don't think I have time to talk about this, but we also have VoucherVision available for you all to test right now on Hugging Face. It has a really easy to use user interface. We let you create your own prompts. So if you don't want to use the 20 fields we can transcribe, you can teach it to do something else instead. You can go to this website on Hugging Face Faces and then just try it out for yourself right now. And we're also making an editor to help with all of this. We don't really trust all these transcriptions right now without some kind of manual check. So we've been developing a user interface to make the process of manual review a lot more streamlined. And we also have, we're building a lot of validation tools such as like GPS location correction and things like that. So this is an ongoing project. I think this is all of our collaborators at the moment. But this is an open project. If you're interested, just let me know and we can definitely rope you into our working group. So if you go to the tiny URL link, there's a little form of interest that you can submit. And then these are all of the links to our project. Yep, thanks.
Thank you so much. Do we have any questions? Wow, okay. We have like 1 minute for questions, so.
So right now it's a GitHub repo and you can run it in a bunch of different ways. So most of the time it's designed for an herbarium to process their locally stored images. So it'd be just clicking a file, giving it a reference point. And then what was the other question about the output? Yeah, so it's meant to do batch processing. So every time it creates a JSON dictionary, it's literally 1 row in a spreadsheet. And as far as we know, most database systems support just uploading a CSV file. So you would need to customize it depending on, if you specify or Symbiota, something like that. But it's sort of just a side step around it. You shouldn't really have to change too much on either.
So in Symbiota portals, there is this duplicate tracking mechanism, and especially as we have so many things across the right, that are being transcribed. That is like one of the most useful options, especially for students or volunteers doing transcription. And I'm wondering if does this tap into that in a way that that same technology kind of like, they just see like duplicate events or duplicate exact, exact matches, certain things like that, instead of actually running it the API.
Not yet. Probably, yeah.
Okay. All right. Thank you so much. Everyone else that wants to talk to William, please find him, talk to him. Okay, our next talk is we have Bahadir Altintas talking about developing an oriented object detection model for museum image datasets.
Okay, I need to share my screen, I think. Okay, can you hear me and can you see my screen? Hello. Oh. Can you hear me, please?
We cannot hear you if you're talking.
And now?
No,
We cannot hear you.
Oh.
Oh, I can hear. Okay.
Can you hear?
Seems that it's on our end. So give us a second.
Oh, you can hear now?
They can hear online, but they cannot hear here.
Oh. Online you can hear, but the audience are not. Let me share my screen again. We are working on that. But can you?
He's not.
I
There's.
cannot hear.
I think we cannot hear here in the room.
Actually, I have a video on the web, Google
Yeah,
Drive if you want. So, because we are also running late.
Oh, we can hear. Success.
Everyone can hear me now? That's okay.
Yeah. Yes.
Okay. Okay. So you can see my screen, I think. Okay. I'm Bahadir Altintas. I'm from Tulane University Biodiversity Research Institute. So I'm going to talk about the developing a fish detection model for museum image data set using yellow weight. So as you can see, cataloging is very important for the biodiversity and the conservative of the specimen. So every museum and every specimen have different kind of pictures and photographs, as well as the text data with them. Here you can see the typical museum image picture, museum image picture data actually. It includes the fish, some scale bar, color bar, some kind of tag or label on it. This is a typical one, but not every time the museum can take those kind of pictures. So from the image data around the world, from the different museums, we have different kind of objects on the picture. Sometimes they have only fish, they have only labels or hand notes. Sometimes they have everything: fish, scale bar, color bar, or label. So we have a variety of data, image data. So our purpose here, actually, we are working on a system which is called Fish Air to prepare AI-ready data providing system. And also we are working on thread extraction from the fish specimen image. So for this purpose, first thing is the filtering the image. So classification is very important here. Which image includes what kind of information in it? Or if, so the question is, every image includes fish or not? Or how many images includes just fish? How many images includes just labels, non-fish, or those kind of things? So for this one, we try to create or train a model for those objects. We chose 5 objects for this purpose: fish, label, scale bar, color bar, and barcode objects. So also we have those kind of information for our some of our data, so we can confirm our model, or we can test our model with those data also. So we chose these object labels actually. So for this purpose, we use almost 2,000 images, and every image is annotated with the objects in it, like fish objects, label and scale bar, barcode, color bar objects. But not every object has the same amount of training data or training annotation or validation annotation. As you can see here, barcode label or barcode object has very few training set sample, and I think we need to improve that one. But all of these annotations done by hand, so it's a time-consuming thing. So we are working on that, still working on these annotations to improve the training. So I used the Ultralytics YOLO 8 model for object detection, and it also supports the oriented bounding boxing model. So if your object is rotated in some angles, you can extract a bounding box. You can extract a bounding box rotation. So when we need a bounding box image of these objects, it will be helpful for us in the future. For test purposes or for time fine-tuning, I trained the model between 50 and 375 epochs with intervals of 25. So to see which epoch is the optimum epoch to use for training. And all the augmentation parameters were used as default from the YOLO 8. So we don't have any extreme augmentations for this. After we trained our models, I think we have almost 14 models with different epochs. We tested all these models on 38K images that we have in hand, and all of those images are annotated for how many fish specimen in that image. We have that information for those images, and if it includes the scale bar in it, or if it has a label on those images. So we have those kind of information for all these 38K images. So it's good for confirmation of the training model to this data set. So we compared the results, or we applied our model on this image data set, and we got results for the fish and other objects with a very good actually accuracy in means of if some objects present in the picture image, and if it's not present in the image, we can check with our results. It's just a presence and absence kind of comparison actually. Also, we have the information of the specimen quantity for the fish objects. So we were able to check the number of objects that our model found from some model, some prediction, and comparing that with the data set we have, which we already know the number of fish specimen in it. So it's like that. You can see the accuracy here. I will show some tables and I will try to explain those. Those are a little bit looks complex, but this table shows on the left side the epochs of the models that I used, and the upper side is the threshold of the score values. So if you look at the first object, it's for the number of fish quantity comparison. So around 0.5 threshold, our model found 98% of the fish correctly. That's the explanation of this table actually. you can interpret this table like this. So we just compare our results with that 38K images manually annotated metadata. And on the bottom is comparison of if a fish object is available in the image, we call it as, okay, fish object contains fish objects true or false kind of comparison here. So we don't care about the number of fish or quantity of the fish here. So for this comparison, we have a higher value of accuracy, which is 99, almost 100% accuracy we can see here. Just checking if there's a fish or not. So our model finds the fish if there is a fish in the picture, regardless of the number of picture specimen objects in there. Also, it's the same thing done by the label detection. If there's a label in the image, so our model tried to find it, or if it failed, you can see the values here. For the labels, it's around 0.15, and but the 90% accuracy can be found in different epochs and different or lower thresholds here. For scale bar, we have over 90% accuracy for 0.2 threshold, and for almost every epoch we can get that results. And the color bar has a 99% accuracy for 0.3, 0.35, or above 0.7. We have 90% accuracy for color bar, because color bar has a very similar structure. For almost every color bar is the same. So we can find or our model can find color bar easily maybe. For the barcoding, it's a little bit distributed. We didn't have enough barcode samples for our training set, but we still have over 85% accuracy for almost every threshold or every epoch's values, every models. In the right bottom, I give the best values, best accuracy along with the threshold and the epoch's values. So our aim is to apply this model to all fish hair data, fish hair org data, which has almost 400,000 images. So I applied this model to that data set, and I roughly get how many fish images available, or how many label available, or how many scale bar information available in this data set. I got those kind of data, or those kind of results roughly. But the main problem here is with the barcodes. So as I said, we don't have enough training set data for barcoding. So maybe we should add those data, and maybe it's confused with another thing, because sometimes barcode or other object can detect it as scale bar or label or whatever. So it gives up rough information about our data that we have in our fish org system. So that's all we have done. I try to be quick, but if you want to learn something in details, I can happy to answer your questions. And FishAir.org is the database system that we are developing now for AI-ready image providing or data providing system. And Tulane University is where we are doing this. And this project is supported by NSF and a part of Imageomics project. Yeah, thank you for listening, and if you have any questions, I will be happy to answer.
Thank you, Bahadir. We have time for questions. If anyone here in the audience has any. Okay, I don't see any questions. Thank you so much for your talk.
Thank you.
Thank you
very
to
much.
all of the speakers of this session. We are coming back at 2 p.m. And again, sorry for all of our technical difficulties, but I think we had a really good set of talks. So we'll see you after lunch. Thank you.